All right, everybody, now that we have the enterics under our belt, woo, we can move on to the non-fermenters. These are basically organisms that do not ferment glucose, and that's why they're called the non-fermenters. So this is Unit 13. So a non-fermenter is a group of gram-negative rods that do not ferment glucose. They are generally asaccharlytic and won't ferment carbohydrates. Some, however, will oxidize carbohydrates. About 15% of all gram-negative rods isolated in the clinical lab are gram-negative rod non-fermenters. These are organisms that are aerobic gram-negative rods and show abundant growth on TSI, but the TSI will be alkaline over alkaline because they do not ferment glucose. They will not acidify the butt of that media. Organisms that are non-fermenters either use an aerobic pathway, the inter-Deuteroff pathway, in the Krebs cycle to attain energy from glucose if they are oxidizers, or they cannot metabolize glucose at all and they are non-oxidizers. So our non-fermenters are either oxidizers, non-oxidizers, or asaccharlytic. 15% of gram-negative bacillus isolated in the clinical lab are non-fermenters. Often they are resistant to antibiotics. Non-fermenter identification can be challenging but fun. You can tell this gal is having a good time. There is a nice flow chart in your textbook for non-fermenters, and you will notice that the flow includes things like results on TSI, whether or not the organism grows on MAC, what their oxidase result is, and what their glucose reaction is. Also, it's a good idea to know their pigments. Let's go ahead and discuss the characteristics that are common to non-fermenters. They are long, slender, gram-negative rods, or they are gram-negative coccobacillus. There is no evidence of glucose fermentation. They're either going to be oxidizers or non-oxidizers. Usually they are positive for cytochrome oxidase result, and usually they fail to grow on MAC, but that's not always the case. Non-fermenters, like I said, can be challenging because they're not as cut and dry in terms of results as the enterics are. These non-fermenters, they are opportunistic organisms, and they are usually found in soil and water, on plants, and in dying vegetation. They are aerobes, and some are strict aerobes, like Pseudomonas aeruginosa is a strict or obligate aerobe. People that are immunosuppressed with something like diabetes, cancer, HIV, or if they've just undergone a transplant are at more of a risk of an infection with non-fermenters. People that have been exposed to traumatic incidences like burns, surgeries, and gunshots are more prone to getting an infection with the non-fermenter. People that have foreign objects implanted in them, like catheters and contact lenses, those are people that are more at risk of developing a non-fermenter infection. And then finally, people exposed to infused fluids like saline, irrigants, IV solutions, nebulizers, and dialysis fluids. These are all people that are more prone to developing a non-fermenter infection. So like I said, non-fermenters are opportunistic, and they can cause basically any type of infection, but some of the infections you see most commonly are wound infections, including burn infections, urinary tract infections, eye infections, respiratory tract infections, and septicemia. Now, cystic fibrosis patients are much more susceptible to a non-fermenter known as Burkholderia cepacia because they have fluid in their lungs, and that is the perfect environment for non-fermenters to grow. And we'll talk more about Burkholderia cepacia in upcoming slides. Again, I'd highly recommend that you take a look at that flowchart in your Mahan textbook. Some common tests used to identify non-fermenters would be the macroscopic pigments and odors. A lot of times these non-fermenters are pigmented, and those pigments get brighter and those odors get stronger at room temperature. We can do glucose oxidation tests, cytochrome oxidase reactions. You may remember that from the Neisseриas when we did the oxidase test. We can determine if the organism grows or does not grow on MAC. We can look at their flagella arrangement. We can determine if our organisms will hydrolyze urea or not. Will they reduce nitrates to nitrites? Will they produce indole? We can do decarboxylation assays, esculin hydrolysis tests, and finally sensitivity tests to penicillin and colistin. Now, the saccharolytic bugs are oxidizers and can split sugars, but the asaccharolytic bugs cannot. Non-fermenters may produce a number of pigments and odors, and a lot of times the pigments will be yellow, red, green, or violet. These pigments and odors can actually intensify at room temperature. So if you ever see an organism that is a bright pigment and has a strong odor, you may be asking yourself, hey, this could be a non-fermenter. Now, sometimes our non-fermenters are oxidizers and other times they are non-oxidizers or asaccharlytic organisms. Oxidation is when you have a utilization of carbohydrates without the use of molecular oxygen. The end product of oxidative metabolism is water and not gas. It's not CO2 gas. So the acids produced in oxidation are kind of weak. So some of our test results could be kind of weak. Oxidizers produce low levels of activity and must have more sensitive medias like molar decarboxylase media or molar media in general. This molar media is a type of media that is very sensitive to low levels of acids. Now remember, enterics always ferment glucose or other sugars, but non-fermenters only oxidize them or they are going to be asaccharlytic. Whenever we're trying to identify a non-fermenter, we want to determine if our organism is going to oxidize glucose or if they're a non-oxidizer. And an initial clue that you're dealing with a non-fermenter is by looking at the TSI. The TSI will have good growth, but it'll be alkaline over alkaline. The butt of the slant is not exposed to oxygen, and if it were acidified, it would actually be an indicator of a glucose fermenter. Because the butt of the slant stays red, we know that we're dealing with a non-fermenter because it doesn't utilize glucose, doesn't ferment glucose. There's a couple methods that we can use to determine if we have an oxidizer or a non-oxidizer. One method we've already done where you use the two OF glucose tubes and we have one that is sealed with oil and one that is not. You can tell if you have a fermenter because the tube that is oiled and the tube that are open will be yellow. An oxidizer, you will see yellow at the top of the open tube because it is exposed to oxygen, and that's where the reaction is taking place. No activity, or when you have an asaccharlytic type of organism, it's going to be bluish at the top of the open tube and green in the oiled tube. Okay, there's also another type of method called the uniglucose tube, and this is a one-tube system where the tube is actually pinched off in the middle, and you can tell if you have a fermenter if the bottom turns yellow or an oxidizer if it turns yellow at the top. Many of the non-fermenters are cytochrome oxidase positive, but not all non-fermenters will be positive for cytochrome oxidase, but most of them are. Make sure that you set up a TSI before making a cytochrome oxidase determination. If your bug is growing on TSI and it's alkaline over alkaline, then you can go ahead and do the oxidase test. You want to make sure that it's not a Neisseria. Remember, Neisseria and Moraxella are also cytochrome oxidase positive, so you want to make sure that you're not dealing with a Neisseria. Our oxidase positive organisms are things like Pseudomonas and alkaligenes. Our oxidase negative organisms are Stenotrophomonas and Acinetobacter. So in summary, most of our non-fermenters are CO positive, but there are a few that are negative. Just understand the most important non-fermenters in the clinical environment are the Pseudomonads, and they are all cytochrome oxidase positive. We also have a few that are cytochrome oxidase negative, and that would be Stenotrophomonas, Multifilia, and Acinetobacter species. Another way that we can get to the gene and species name of a non-fermenter is by determining if the organism grows on MAC. MAC is not only for enterics, but some of the non-fermenters will grow on MacConkey agar as well. All enterics grow on MacConkey, and many other gram-negative rods will not. So our Neisserias and Moraxellas will not grow on MacConkey. So MacConkey is basically selective and differential for enterics and some non-fermenters. If you have an organism that fails to grow on MAC, we might want to think it's a non-fermenter. But some non-fermenters do grow on MAC. These are not cut and dry like the enterics. The nitrate reduction test is a test that we've done a couple times, and it is a very nice test for the identification of non-fermenters. Some non-fermenters will reduce nitrates to nitrites. Some non-fermenters will reduce nitrates to nitrogen gas, and some will not reduce nitrates at all. So that's a little bit different than our enterics and our staphs. Our staphs will reduce nitrates to nitrites, and so will our enterics, but our non-fermenters are kind of all over the place. Some will ferment some will reduce nitrates to nitrites, others will reduce nitrates to nitrogen gas, and others will not reduce nitrates at all. So following the addition of alpha-naphthylamine and sulfonylic acid in a nitrate broth, we want to see if it turns red. If it turns red, it's positive for nitrate to nitrite reduction. If the red color in the tube is not observed after a 5-minute waiting period, then we add zinc dust. If the zinc dust turns pink, it means the nitrates are still present and have not been reduced. If it turns colorless with the zinc, it means that nitrates have been reduced to nitrogen gas. So both of those are considered positive nitrate tests. Now, it might be important to understand what type of flagella each one of our organisms have. So organisms with flagella are motile, and we can use a flagella stain. We did not do the flagella stain in micro because the flagella tend to fall off and you need greater magnification to see them, but I want to talk a little bit about the flagella arrangement on certain organisms. Okay? Polar flagella is also known as monotrichous, where you have one flagella at one end. Lophotrichous is when you have more than one flagella at one end. Amphitrichous is when you have one or more flagella at each end. And paratrichous is when you have flagella that surrounds the entire cell. Okay, now there are some nonfermenters that are non-motile, one of which is called Acinetobacter, and another one is called Elizabethingia. Those are both non-motile. I want to talk a little bit about TSI and KIA reactions when it comes to the nonfermenters. KIA is Kliegler iron agar, and it's very similar to TSI, except for TSI has 3 sugars, lactose, sucrose, and glucose, where Kliegler iron agar only has 2, lactose and glucose. Now, some very fastidious organisms fail to grow on TSI entirely. They won't grow there. Nonfermenters, on the other hand, will grow on TSI, but because they are asaccharidic or oxidizers and not fermenters, the slant will remain alkaline over alkaline. Some nonfermenters produce pretty stunning pigments. For example, Pseudomonas aeruginosa produces this beautiful green pigment. Anytime we're dealing with a pigmented organism, you should be considering that it is possibly a nonfermenter. Pseudomonas aeruginosa produces the pigment pyocyanin, which produces the green pigment. So anytime you see the green pigment like what is noted in this image here, it has to be a Pseudomonas aeruginosa. Other pigments you may see are yellow, red, and purple. Elizabethingia is a yellow organism. Methylobacteria is a red organism, and so is Micrococcus roseus. And then one purple organism that we've dealt with is Chromobacterium. Those are because of the pigments phenazines, which is a red pigment, and violacine, which is the purple pigment. These colors will intensify at room temperature. And some people like to do microbe art. So here's a picture of, I guess that is a duck or a swan with a little tree branch there. This happens to be Shingomonas possumobilis, which is a yellow pigmented organism. And it's just kind of cool. Nonfermenters tend to have strong odors as well, and those odors will intensify at room temperature. Some of the most smelly organisms are nonfermenters. These odors are going to intensify at room temperature along with their pigments. The most noted nonfermenter odor that occurs is for the strain of Pseudomonas aeruginosa, and it smells like grapes, and to some people it smells like corn tortilla chips, and sometimes a certain strain of Pseudomonas aeruginosa will smell like moldy grass. Alkaligenes faecalis smells sweet, and Myroides odoratus smells like green apple. Here's a list of some of the nonfermenters that we're going to discuss in class. The ones that I really want you to study hard are the ones that are highlighted in red. Pseudomonas aeruginosa, Stenotrophomonas multifila, and Acinetobacter baumannii wolffi. Those are the 3 nonfermenters that are seen most commonly in clinical laboratories. Pseudomonas aeruginosa by far is the most common one you're going to see, followed closely by Stenotrophomonas and Acinetobacter. Other ones on the list would be Pseudomonas putida, Burkholderia cepacia, Alkaligenes xylose oxidans, Olagella ureolytica, Shingomonas possumobilis, Alkaligenes faecalis, Olagella ureolytica, oh, I got that on there twice, Brevundamundus diminuta, Provundamonas vesicularis, and Shewanella putrefaciens. I kind of like Shewanella putrefaciens because it's the only H2S positive non-fermenter. Now, some of the tests that we can use to identify non-fermenters would be things like our macroscopic description. What is the pigment and the odor of the organism you're trying to identify? We can do OF oxidizer or non-oxidizer tests. We can determine if our bug has grown on MacConkey or not. We can place it in different temperatures and see where it grows optimally. We can do the cytochrome oxidase test. And then other tests we would do would be indole, urea, esculin, and motility tests, nitrate reduction tests, and finally we can see if our organism is sensitive or resistant to penicillin or colistin. This is just kind of a condensed list of all the tests that we do to identify the non-fermenters. Pseudomonas aeruginosa is the most frequently encountered non-fermenter in the clinical environment. It is found in moist environments and especially in burn units. It can cause things like burn infections in swimmer's ear, cutaneous wound infections, respiratory infections, keratitis, which is an infection of the eye, and urinary tract infections. I've actually seen people with urinary tract infections where their urine has been bright green like the pigment of Pseudomonas aeruginosa. The virulence factors of Pseudomonas aeruginosa would be proteases and exotoxins and also extracellular slime. That of course is your glycocalyx. Minimal definitive identification requirements would be that it's cytochrome oxidase positive, it has a strong odor and pigmentation, that it's going to be green, and that it's a gram-negative rod. Pretty much in the clinical world, if you see a green organism like the one on the bottom right, if it has a metallic sheen on blood, is beta hemolytic and oxidase positive, you can pretty much call it a Pseudomonas aeruginosa. Pseudomonas aeruginosa is in what's known as the fluorescent group of non-fermenters. Non-fermenters either produce a pigment known as pyocyanin, which is a greenish pigment produced by most strains of Pseudomonas aeruginosa, or pyoviridin, which is the pigment that allows the organism to fluoresce under ultraviolet light. Characteristics that are common for Pseudomonas aeruginosa: on blood agar they have a metallic sheen. It almost looks like tinfoil on blood. They are beta hemolytic most of the time and produce a green pigment. Now sometimes they can be kind of brownish or bluish as well, but if they produce that classic green pigment, then we know it's a Pseudomonas aeruginosa. They are oxidase positive, they're glucose oxidizers, and they grow on MAC, but of course they would be colorless on MAC. They prefer 42 degrees. They grow best at 42 degrees, even though they will grow at 35 degrees. The TSI is alkaline over alkaline because they don't ferment any of the sugars. And then their lysine and ornithine decarboxylase is going to be negative. They do happen to be arginine decarboxylase positive, however, and they will reduce nitrates to nitrites. It can be difficult to treat Pseudomonas aeruginosa because certain strains can be more resistant to antibiotics. They are usually sensitive to the aminoglycosides, including gentamicin and tobramycin. They're also susceptible to the carboxypenicillins, and they are usually susceptible to the third and fourth generation cephalosporins such as ceftazidime. A lot of times they'll give two antibiotics together that work better together than they do apart. They'll use them synergistically. One such example of that would be the use of timentin and cefepime at the same time to treat Pseudomonas aeruginosa. And here's another interesting picture where we have the green color of the Pseudomonas aeruginosa, and then if it's causing an eye infection, what do you suppose that would look like? It would look like this. You can see that green pigment in your patient's eye. Other Pseudomonas I want to talk about is Pseudomonas fluorescens and Pseudomonas putida. These are both part of the fluorescent group, even though neither one of them produce the green pigment pyocyanin. Okay, these are rarely encountered, so you don't need to know too much about them other than they are in the fluorescent group. Then we have Pseudomonas stuteri. It's found almost everywhere in nature. It's rarely the cause of human infections, but it's very interesting and recognizable, and I'll show you in the next slide what that looks like. So Pseudomonas stuteri is easily recognizable. Once you see it, you'll never forget it. It almost looks like dead skin or blistering skin on the media. Burkholderia cepacia complex is an opportunistic organism, and people that have cystic fibrosis often develop lung infections with Burkholderia cepacia. They develop what's known as cepacia syndrome. It may be difficult to identify this organism because it contains 9 genetic species. It's not just one single phenotype. This particular organism can be yellow. It's going to grow on a selective media known as Burkholderia cepacia selective agar, or BCSA. That would be the media of choice to isolate Burkholderia cepacia. The organism actually turns pink on that media. They are a weak cytochrome oxidase positive. It's a glucose oxidizer. It's OMPG positive, which is interesting. It is actually a slow lactose fermenter, but we classify it in the non-fermenter category because it is such a slow lactose fermenter. It's urea positive. It's going to grow on MAC. It will not grow on Salmonella or Shigella agar, which is another media we can use to identify our non-fermenters. And they are negative for nitrate reduction. And Burkholderia cepacia is also resistant to penicillin and colistin. Okay? If for some reason your cytochrome oxidase test is a little bit too weak, you can try using the swab method. Okay? Now, when it comes to identifying some of these non-fermenters, it might be best to use commercial kits like API, which stands for Analytical Profile Index, REML identification using the RAP-ID method, or Vitek. They're all getting better at identifying these non-fermenters. Stenotrophomonas multifida is pretty commonly isolated in the Midwest, especially in respiratory cultures. It is unclear if it's a pathogen, but it is opportunistic and looks and smells similar to Serratia. It's generally multi-drug resistant. It's very recognizable in its antibiogram. Its antibiogram is basically what does this organism look like in relation to the antibiotics, and you'll be able to see that it's very resistant. On McConkey, it grows. The oxidase is going to be negative for the most part. It's a glucose oxidizer, will reduce nitrates to nitrites, and is lysine decarboxylase and ornithine decarboxylase positive. Your arginine is negative, and it is modal. Acinetobacter species is a non-fermenter that is a gram-negative coccobacillus. It is actually non-modal and cytochrome oxidase negative. Now, I've gotten burned on this organism before because on McConkey, it looks like a sickly E. coli or an E. coli that's been treated with antibiotics. There are several genome species within the genus Acinetobacter, including Acinetobacter baumannii, Acinetobacter woofii, and Acinetobacter chalcosidicus. Usually we report these out as Acinetobacter species or Acinetobacter chalcosidicus baumannii complex. Okay? This can be found in the respiratory tract of certain humans, and it can be an opportunistic pathogen. It can also lead to urinary tract infections and wound infections. It can be normal flora also of the skin, respiratory tract, and of the gut. So all of the other non-fermenters that we've talked about so far are gram-negative rods. This one happens to be a gram-negative coccobacillus. Okay? It is cytochrome oxidase negative and non-modal. It's a strict aerobe and does not reduce nitrates to nitrites. It may demonstrate high antibiotic resistance, but that really depends on the strain. On McConkey, it can look a lot like a sickly E. coli. And then the OF glucose is variable. Acinetobacter woofii is a non-oxidizer, while Acinetobacter baumannii is an oxidizer. So here's a picture as to what I was talking about. A lot of times Acinetobacter will look like a pale pink E. coli. Sometimes if you're treating E. coli with antibiotics before the culture is collected, your E. colis can look like this also, where they just kind of look sickly. Acinetobacter looks like a sickly E. coli. Alcaligenes faecalis is one of the most commonly isolated species in the genus and is the one we're going to focus on. The old name was Alcaligenes odorans. Some species produce a strong green apple odor and start to green the agar. And what I mean by that is not that the organism is green, but it starts to get, it almost looks to be a little alpha hemolytic around the colony. And that's not alpha hemolysis. What that is that the organism is just using the nutrients in the media. Now, alkaligenes species can reduce nitrites rather, but not nitrates. So that's kind of interesting about alkaligenes. It is an opportunistic pathogen. It can cause ear infections, urinary tract infections, and septicemia. It's also a contaminant of water or wet environments such as swimming pools, dialysis fluids, and tap water. It is oxidase positive, will grow on MAC. It's kind of variable as to whether or not it'll grow on Salmonella and Shigella agar. It is a non-oxidizer of glucose and may strongly alkalinize the media. Urea is negative and it is tolerant of 6.5% salt. Here we have two different types of Achromobacterium species that we're going to talk about today. Achromobacter denitrificans and Achromobacter xylose oxidans are the two species I want to talk about today. Pretty much these two species are identical with the exception of the OF glucose test. You can see that one is a non-oxidizer and one is an oxidizer, but they are both cytochrome oxidase positive and they both reduce nitrates to nitrites. Elizabethingia meningioseptica was formerly known as Flavobacterium and Chrysiobacterium. Okay, this was named after an American biologist, Elizabeth O. King, who worked with a yellow pigmented gram-negative rod associated with meningitis in infants, possibly Coronabacterium sakazaki. It's hard to say. Some organisms may cause wound infections and septicemia. It is variable on McConkey. Okay, depending on the serotype, it may or may not grow on MAC. There's no growth on Salmonella and Shigella agar. It is a weak glucose fermenter, so weak that it's treated like a non-fermenter. It appears to oxidize, actually. It is cytochrome oxidase positive, indole positive, and urea negative. It will not reduce nitrates to nitrites, okay? The indole reaction will be positive, but it's going to be very weak with Kovacs, and so what we'll use is Ehrlich's reagent, which is just a little bit more sensitive. And then also Elizabethingia is sensitive to penicillin and resistant to colistin. Okay. In March of 2016, there were 44 cases of Elizabethingia related to septicemia, and 18 of those 44 people died. Here I want to talk about Bordetella, but I'm not going to talk about Bordetella pertussis too much at this point, but we will get to that when we talk about the respiratory pathogens. Okay, Bordetella, there are 7 species of Bordetella. It's a gram-negative cocci at 24 hours, becoming more pleomorphic at greater than 24 hours. Pleomorphic means that in a pure culture, you see a variety of shapes and sizes to the individual cocci. Humans are the only host for Bordetella pertussis. That is the organism that causes whooping cough. But Bordetella bronchoseptica is mostly found in animals like dogs and pigs. Okay, the syndrome begins with a cathartal stage or flu-like symptoms when you have Bordetella pertussis. The second stage of Bordetella pertussis is the spasmodic stage where you have coughing spells and a whoop is heard at the end of the cough. There's periods of hypoxia and vomiting when you have Bordetella pertussis. Hypoxia, of course, is inadequate perfusion at the cellular level. And let's move on to the one that is associated with dogs and pigs. Bordetella bronchoseptica is the one that is related to dogs when they get their Bordetella vaccines. This is mostly isolated in animals. It can cause an opportunistic infection in humans. It can lead to sinusitis, pneumonia, and septicemia. But it is Bordetella bronchoseptica that causes kennel cough in dogs and cats, which may be transmissible to humans. And it also causes a severe respiratory disease in swine. Phenotypically, Bordetella bronchoseptica is quite different than Bordetella pertussis, but Bordetella bronchoseptica can lead to severe respiratory diseases not only in dogs but also in humans and pigs. This organism will grow on MAC. It is catalase and cytochrome oxidase positive, and the key reaction is that it is very rapidly urea positive. It will reduce nitrates to nitrites, and it is a glucose non-oxidizer. So, this is an introduction to the non-fermenters. They are difficult to identify compared to the enterics. When you get out to the clinical world, most of the time they're going to let you use a flowchart to identify these non-fermenters, or they're going to let you use a rapid method like API, microscan, or Vitek to identify these organisms. We'll do a little bit with these organisms in lab, but we're not going to do a whole lot.